this technology. Now, this is another example from developing an industry-oriented development. So a corn that is called waxy corn that has been developed by DuPont Pioneer and Corteva AgriScience. So this is if it has more amylopectin, so it becomes very waxy in nature. And this waxy corn is very important in industry for processed food and making adhesive and high gloss paper. So if you see a starch biosynthetic biosynthetic pathway, so the sucrose comes here to ADP glucose, and ADP glucose go into two directions. One is amylose, another is amylopectin. So now if you just inhibit the gene which is synthesizing amylose, then sorry for the interruption.
When you have to work from home, sometimes you can't control what happens.
Hello, I'm sorry, somebody is working in our rooftop, so in order to reach him, to stop him, so I have to stop here for 10 minutes. So I think it is not viable to stop presentation here. So I'm just continuing. He would be stopping soon, I think. Okay. Okay, so what I was discussing about this waxy corn, this is very valuable for these industrial applications. So if you inhibit the amylose production pathway, then you channelize the ADP glucose towards the amylopectin synthesizing pathway. So then you get a lot of amylopectin. So they developed these corn with more than 97% of amylopectin by deleting the waxy gene that is responsible for amylose synthesis. This is another development. This is, if you see a mushroom, if you cut a mushroom and if you keep it for a longer time, that this mushroom, the tendency of that mushroom to become brown and black, okay? And if it becomes brown and black and nobody likes it and nobody would like to buy it. So what has been done by Pennsylvania State University scientists, I was working with the same lab, in Ong Yang's lab, and what they have knocked out the PPO gene, that is polyphenol oxidase gene, and this polyphenol oxidase actually causes the browning of the at the cut end by oxidizing the secreted phenolics. So when they knocked out this PPO gene, then they could able to achieve to develop a mushroom which would not brown as fast as the original one. So this is called non-browning mushroom by applying the CRISPR-Cas9, and it's very rapid. So this is another very interesting example in case of reducing or eliminating a health problem due to ingestion of some food. So if you eat wheat, barley, and rye, many Western country people, they have this problem of celiac disease and they are intolerant to gluten. So, if you go to any meeting, so I always used to see this is gluten-free bread, this is gluten-free, all these kind of things. So, because some people, if they eat that food from wheat, barley, or rye, they have some problem in their intestine. If you see what happens to them, so this is a normal small intestine, and this villi is responsible for absorbing nutrition from the food that we intake. And now, if you continuously eat this gluten, this wheat, actually this wheat contains a protein that's called gluten protein, and this is made up of gliadin and glutenin. This gliadin actually is some small intestine of some people, they recognize this gliadin as an antigenic one. So they develop autoimmune response because of ingestion of this gliadin. One second. Okay, I was telling that this gliadin is actually recognized by immune system as a foreign body, as an antigen. So they develop antibody against this gliadin. So because of this autoimmune response, the villi became destroyed, and it looks like this kind of destroyed villi. And when you have a destroyed villi, then you won't be able to absorb nutrition as you do in normal condition. So this is when a person develop the celiac disease, and this is because of the problem of gluten. So now the problem is, the question is, do we have a tool to selectively remove or selectively disable this gliadin forming gene? Hello. Yeah, I'm back. I'm sorry to the audience for this inconvenience. I directed someone to look after this one. Okay. Okay, so this is an example of developing a low gluten wheat with CRISPR-Cas9. So if you see the gliadin gene that is actually causing the problem for this gluten allergy. So this is the red highlighted one, which is the portion that is producing an epitope of that antigen. The gliadin, some portion of that gliadin protein is actually recognized as an antigen, and that is called epitope. So this epitope is formed from this sequence. So what they have done, they designed this guide RNA and to disable that portion especially, and they developed low gluten wheat by using, this CRISPR-Cas9. So if you see this graph, this is multi-top graph, this is the gliadin fragment, extracted gliadin signal, whereas this one, blue and this yellow peak is the one from the edited wheat by CRISPR-Cas9. So you can see greatly reduce the percentage of gliadin in the edited plant. And if you see the phenotype of this one is the control one, which is not treated with CRISPR-Cas9, and this one is the edited one, which is developed low gluten CRISPR-Cas9 mutant. So this looks exactly similar as the original one, but it has less gluten than the wild type one. So this is a great example how CRISPR is impacting to remove the anti-nutrient factor from our crop. So this is another similar kind of example of caffeine. The problem is caffeine. So we manually do decaffeinating our coffee. But if you could develop a plant which would produce decaffeinated coffee, that would be great, okay? And that is possible because of CRISPR-Cas9. So this is a caffeine forming or caffeine synthesizing pathway, and they're targeted. This is Tropic Biosciences working from UK. They are working on this one, and they have knocked out, and they're trying to knock out with that the theobromine synthase gene to inhibit the synthesis of caffeine in the coffee plant. So we would soon have a coffee plant which is naturally decaffeinated. So you would save a lot of money. You don't have to go for decaffeinating your coffee after harvest. Okay. So this is really humanitarian work is going on in UC Berkeley. Innovative Genomics Institute is the institute from, Cassava is the staple food for over 800 million people all over the world. And it is the major source of calories for roughly 40% of African people. And they are mostly poor. And, because they don't process it, and a lot of they don't have all the resources to process it properly, then they eat very raw kind of things, and they don't follow all the proper procedure. So these cassava actually contains a toxin, cyanogenic glycosides, and that's toxic to human. If you consume regularly, means year after year, and with very less protein containing diet. So they are very poor, and they only depend on cassava, and they are eating it raw and not properly processed, and don't have any protein in support. So they develop irreversible paralysis in their mainly in children and mainly in children and in children and women. So this disease is called Konzo, and this Konzo, in order to remove this Konzo from the African nations, Innovative Genomic Institute, Dr. Jessica Lyons and this Myung Zeso, they are working on this one to knock out 2 genes that are actually responsible or that catalyzes the formation of that cyanogenic glycosides. So if we have a cassava who does not have that toxin, we could save a lot of life and from suffering from Konzo. So this is under development. If I want to give you a snapshot of how it is impacting in crop protection, this is one slide. So a lot of pathogen affects our plant, and some plants are susceptible and some plants are resistant. Because some plants are more vulnerable to a disease, to a pathogen, to a pest, because the plant has susceptibility genes. Actually, those genes is plant's own gene, but they help in pathogen establishment, and they modulate the host defense, and they also help in pathogen sustenance by giving them food and all this kind of stuff. This is called susceptibility genes. So now, if you see a classical example of the interaction between bacterial blight pathogen, that is Xanthomonas oryzae, and the rice plant interaction, what they do, this Xanthomonas bacterial blight pathogen, they secrete a protein that's called tail protein. And this tail protein goes and bind with promoter of certain genes. And that gene is a susceptibility gene that helps in establishment of pathogen. So how they do that? This tail protein, when they bind to the promoter, then they increases the expression of the gene. So here the example is a sweet gene. Sweet gene is nothing but a transporter. So it's a protein. This gene produces a protein that transports sugar from nuclei, sugar from a cell to the apoplastic region, intercellular space. So now, when this transporter is produced more by the influence of the bacteria, because bacteria is influencing the more expression of sweet gene, then the sugar is transported more in the apoplastic region, and the bacteria reside in the apoplastic region, and they have more food. They're enjoying more sugar, which is produced from the plant. So they are tricking the plant's own system to have more food. So this is a classical example. And because we know the basic biology, then we could interrupt specifically the promoter of that gene by CRISPR-Cas9 to inhibit the binding of tail protein with the promoter. So if you say, these are the 2 results from Nature Biotech. One is CRISPR-Cas9, another is another genome editing technology, Talent, and these are the results here. So when they changes the binding sequences of the promoter specifically, then the tail would no longer be able to bind with the promoter. So the sweet gene would not be expressed in a higher amount. So the bacteria would not be benefited. Okay, so this is the strategy how CRISPR-Cas9 is being used for developing pathogen-resistant crop. This is an example from rice, but a lot of other things like citrus, canker, and also powdery mildew resistant wheat, like by deactivating MLO genes and all those kind of things. So I'm not going into details because I don't have time to accommodate all those details here, but you could, if you are interested, you could go through my other lectures. Now, another question. Till now, we have learned how CRISPR-Cas9 can disable a sequence, can disable a DNA, can disable a gene by introducing random insertion and deletion, which is not in our hand what insertion and what deletion would be there. But here is an example from a plant. You see, this is a Japonica rice variety, which has a BSRK gene. It's called broad-spectrum resistance Cuttack gene. And that gene has a single nucleotide polymorphism. If it has a C in a position, C letter, or cytosine, then it is more susceptible to a blast pathogen. And when it has a T letter in the same position, another sequence of the gene is same, only a single letter is changed to T, then it becomes resistant. So this is a great example of how one single nucleotide can change the appearance and can change the response to a foreign invader like pathogen, okay? So this is how C and T difference makes huge difference in disease susceptibility and resistance. Now, the question I want to ask you, is there a way to rewrite genetic letters precisely that you want to change from C to T? The answer is yes, only after the invention of this base editing tool. There are two kinds of base editing tool. One is cytosine base editor, that can cause the C to T mutation, that can change a C letter to a T letter. Another is A to G. You can make with adenine base editor that can change an adenine to guanine. So probably you remember from my earlier slide that I introduced you to Cas9 that has one caesar instead of two caesar, okay? So this base editor has been developed with Cas9, that is Cas9, and fused with deaminase enzyme. So I'm not going into details how they do all this kind of stuff, not into the basic technology, how they move, but you have to go through my other presentation to understand better how they actually changes selectively one base to another base. So this is an example from my work at Penn State. So we targeted this WSL5 gene, and we wanted to see whether we could also change the single nucleotide in a very precise manner to another nucleotide that we want to change. So we know from our literature that in this WSL5 locus, if this A is changed to a G, then it develops a phenotype which is easily detectable, white striped in the leaf. The leaf is normally green, but if you change this single A to a G, then it changes its color to a white striped leaf. So we tried with that one with adenine base editor. We developed adenine base editor for plant system, for rice especially, and we targeted this A, and you can see this is amazingly, we could convert this targeted A to a G nucleotide specifically. You see, this is a chromatogram, and this chromatogram means green peak designate the A, and black is G, blue is C, and red is T. So you can see this green peak has been converted to a black peak. That means A has been converted to G specifically by the application of adenine base editor. But you can see the other peaks, they all remain unchanged. They have not been changed. So I can specifically, in a targeted fashion, change one single nucleotide to another type of single nucleotide by these tools. And you can see this is the altered characteristics, altered phenotypes that we achieved in the mutant. So it is clearly visible that single nucleotide change can cause this kind of phenotype, and we can achieve genotypically similar kind of change with this base editing tool. Similar with our second target, and we have targeted more other genes, but I'm just giving you these two examples. Here, it is called zebra3 locus or Z3 locus. Here, we target this A, and similarly, we could convert this A in a targeted fashion to a G in the mutant plant. And others are not changed, okay? And here we have seen similar kind of phenotypes that is expected due to the single nucleotide change. And we have achieved it with the in a multiplex fashion. In a single experiment, we did this 2 targeting and we achieved both kind of phenotype. So this is now coming to an application-oriented application of base editing. So in crop production, how it is helping, 30% of the what we invest in agriculture, 30% goes to manage weed, especially in developing countries like India and all those Asian countries. Most of them, they do, manual weeding. So those labor, they comes and they pluck weed from the field by their own hand. And a small instrument also they use. So this is manual weeding, but almost 30% in global scenario, 30% is gone to this weed management. But this base editing could develop a herbicide-resistant plant development. So if it is herbicide-resistant, so you could go for chemical weeding, So the weed would be killed, but the plant would not be killed because it is resistant to that chemical. So here, this is a target, CGC. If the C is changed to a T, then the amino acid alanine is changed to a valine. And this could be done with cytosine base editor because C to T change, okay? And these single amino acid change in ALS gene, acetolactate synthase gene, could give you IMZ resistant plant development. So this is one example. Another, this has been used with adenine base editor, and they targeted another gene that is called OSACC. This ACC gene is actually target of another herbicide. When you apply herbicide, it goes and kill a protein. So that protein is very essential for the plant's own survival. So how it kills the plant, okay? Now, if you apply that chemical and the plant dies, and if the plant becomes resistant to that chemical, then you develop a herbicide tolerant plant. So here, what they used adenine base editor to change this cysteine to arginine by changing A to G in the opposite standard is T to C, and they could develop a herbicide tolerant rice plant applying adenine base editor. So here's another, this is another fascinating area in plant science, and only 2 papers are available, but in other cases it is being used. This is called directed evolution. So you could change the sequences of a target gene, and you could enrich the sequence, and you could evolve artificially a gene of interest if you want. And you could develop novel kind of mutations, and it could give rise to a novel character that you never imagined. So this is a classical example from a group from Saudi Arabia. This is a chemical. This is called splicing. Splicing is RNA splicing. That's intron, exon. So in normal eukaryotic RNA, the intron is spliced out and exon retains, and then it codes for protein. Okay. So this is normal cases. For it is a very fundamental procedure of any biological system, means any eukaryotic systems. Now, if you see, this SF3B1 is a gene that is a very important component in a spliceosome or splicing machinery. Okay. Now, what they have done, this group, they designed a lot of different targets. These are all different guide RNAs, okay? So they designed different targets all over the gene sequence, and they made a library of that construct, and they transformed. It means they made cut at several positions, okay? And then they go for direct screening. So you get a mutant population of that gene, targeted mutant population. It's not that gamma radiation, the whole genome is mutated. It's not like that. It's a targeted mutation, directed. That's why it is called directed. So directed mutant population of that gene. And then you select for a splicing inhibitor chemical, that is GEX1A. And in presence of that chemical, what they have seen, that one kind of mutant is surviving at a very high percentage of that chemical, but other, including the wild type, that could not survive in that chemical. So what is the outcome of that study? You see, this splicing inhibitor chemical now comes as a new herbicide that was not known to the plant society. And this herbicide could be used, and this herbicide tolerant rice plant could be used to better manage the weed, because if you would not die because they have the resistance power. Okay. Hello, am I okay? Yeah, okay, good. Now, this is another example of using dual base editor. So I talked about cytosine base editor and then adenine base editor. Now what they have done, they developed a new base editor which has both the properties, so it could perform C2T and also A to G editing by a single construct. So they have also targeted another gene that is called ACC, and this ACC gene, when they have in a similar fashion using the dual base editor, they have developed a lot of mutant population, and then this population, and they sprayed with the known herbicides, and they have seen some plants are surviving. And when they have genotyped that plant, they have seen novel mutations is giving resistance to that herbicide. So it is improving the basic understanding of how herbicide resistance develop, and also you could come out with new novel kind of herbicide tolerant plant that could be used for better weed management. Okay. Now this is almost the last part of my presentation. So how machine learning is a fascinating area. Everybody talks about artificial intelligence, machine learning. You know, this machine learning has a lot of application in CRISPR field, because we are, I was a part of that team in Inong's lab for predicting CRISPR-Cas9 induced mutation for precise editing. So I told you that CRISPR-Cas9 normally makes a double strand break, and then the repair system develops random mutation, random insertion and deletion, so that it is the gene is non-functional. But now people are telling that CRISPR-Cas9 outcome could also be predicted. So what kind of insertion, what kind of deletion would be there could also be predicted by applying machine learning. So what we would do that, if we have a weak repair data, so we could feed that repair data with our experimental details, what we have used during our experimentation, and we could develop a machine learning. And from that huge amount of data, they could develop a model, and that model would predict which guide is giving you the best percentage of knockout mutation, and which guide is more efficient, and which guide is going to produce what kind of mutation. Those could be predicted by using this predictor models. So it is still in the process and painstaking. So let me give you a simple example by here. This is a 20 base pair target guide RNA, and if the fourth position from the PAM sequence is T, and if you apply Cas9, so you have a high chance of insertion of another T. So this is our prediction says. And we have seen in 2 or 3 of our experiment that if I use this kind of guide, we have maybe 40% of time we get another T inserted. So this is good prediction using machine learning. So this is another example just a few weeks ago came. So it says machine learning, they have utilized machine learning to predict the outcome of base editing, base editing. Right? Base editing means adenine base editor and cytosine base editor, okay? So what kind of single base editing you are going to get, this would give rise if you use these machine learning tools. So now this is the last part, and one of the most important part when